Good morning, everybody. So thank you for your introduction. Today I want to talk to you about reprogramming approaches to study aging and genetic risk for vascular disease. So this is probably a very simple slide that show you two big areas of interest on using iPSC. On one side, we have basic and translational research, so we can study physiological and pathological mechanism, doing disease modeling and probably drug discovery. And on the other hand, we have, of course, regenerative medicine. So iPSC can be used for transplantation, personalized medicine, and cell banking. So today, I want to just share with you two stories from our lab. One will be probably from this side of the panel about regenerative medicine and how we can use safely iPSC. And the other part of the talk will be disease modeling, and in particular, just how we can model genetic risk for disease. As you all know, iPSC are already used in the clinic. This was the first transplant of iPS-derived retinal cells on a Japanese woman who was 70 years old and treated for a macular degeneration. So this was very exciting, and actually a few months ago it was published a follow-up of this study. The cells are still there, so the patient didn't progress in the disease, so I think this is very promising. But the problem is the second patient of the trial couldn't receive the iPSC. So the trial was canceled because the iPSC contained mutations that were not found in the original cell. So, and these open a new questions, right? So what is the cause of mutation in iPSC? So we can find mutations that are de novo mutation acquired during reprogramming or culture, as in this case. And of course, these are not going to be 100 penetrant in the population. So we can kind of clear them out or just find new methods that are more efficient for having better iPSC. But what is more interesting, at least to us, is we are going to find also rare somatic mutations that are present in the original cell. In this case, they're going to be rare, so in one probably cell. But the problem is when you do iPSC, you go from one cell to one line. So in this case, your mutation will be 100% penetrant. So the mutation will be carried through the iPSC line. And what is the cause of this mutation? Of course, one of the major causes of somatic mutation is aging. So as we age, in time, our cell can acquire somatic mutation. And this is a problem if we think that we want to make iPSC from old donor, like the 70-year-old woman. So we need to face the problem of aging in iPSC. And if we want to study aging, of course, we need also aged sample. So what is the best collection of samples you can have if you want to study aging? So we got very lucky to collaborate with Dr. Eric Topol and Ali Torkamani at TSRI. And we were lucky to use this very nice collection. It's the Welderly. So those are a collection of blood samples from people that are between 85 and 100 and more years old. So they are completely healthy, no sign of major diseases, not aging-associated disease. So these will be probably the best control to use. So healthy people with no aging-related disorder. So our question is how the influence, how the donor age can influence the iPSC quality. So for our study, we took the elderly samples, but also donors of all ages from 20 up to 100. We took their blood and take PBMCs and expand them and reprogram. And for reprogramming, we chose to use 3 lines per donor. And we did a very simple study. We looked at the methylation state and also at their genome. So we did DNA methylation analysis and exome sequencing. What about the methylation state? So we use this ORVAT method. It's a way to predict age on tissues. It works pretty well, and we tried it on our PBMC population. And as you can see, you can predict the age in a very accurate manner. So the error is probably plus minus 1 year, so you can predict the age of the donor just from the blood sample. We applied the same method to our iPSC, and as you can see, the iPSC have a negative age, so they are kind of gestational age, right? That I think makes sense, but we can see still a kind of signature of age. So we can see iPSC from old donor looking older than iPSC from young donor. So then we can actually detect age in iPSC. And we look at the whole methylome, and we found something very interesting. What I'm showing you is just about 33 CpG sites. I think I found them very interesting because, as you can see, these loci are very methylated in the PBMC's population, and they are supposed to demethylate completely during reprogramming in the iPSC. And this is what happened with iPSC from samples at young age. But as you can appreciate, in the middle of this heat map, so there is more yellow, right? So the problem is the aged iPSCs have a little bit more methylation. So what we found is that actually we can see signature of age in iPSC, and this signature, this memory of age, is due to a resistance to demethylation at some loci. So this can be erased, mostly, not totally, with passaging. So if we passage the cells enough time, we can do the same methylation analysis, and we find that we can almost erase completely this resistance to demethylation. So methylation can be fixed in some way. What we cannot really fix is the genome, because what we found is that so with age, the somatic mutation rate actually increases a lot. So you see a linear correlation with age, so you can find more somatic mutation in old donor. Except for this weird 100-year-old sample, it was a kind of a reduction in somatic mutation. So what we did was going on and reprogram more cells, spanning age 90 to 100, and to see if this reduction was actually true. And indeed it is. It's very interesting that very old donor actually have way less mutation than expected. And this is something we can discuss later probably. But what is interesting is what happened is during aging, so the blood can accumulate unique somatic mutation, and this will be transferred to the iPSC. So of course, each iPSC can have different mutation, so you'll have different potential problem for safety in each single line. So, and this is just an example, is just a table of few mutation we found in our iPSC line. And these are predicted to be loss of function mutation. They are in genes that most of the time are important for disease. So what we want to know is where our mutations are and if they are impacting disease-causing gene, because this is going to be an issue if we want to do transplantation. So to conclude the first part of my talk, I would say we can reprogram blood quite efficiently from any age. And I think if we are thinking about reprogramming very old donor samples, maybe blood will be a very good tissue to reprogram, since we know that fibroblasts sometimes can be hard to reprogram if they are from old donor. But the iPSC can retain an epigenetic signature of age. This can be erased, but it's something that we need to think about for sure. And the somatic mutation increase over time, so it's something that we want to think if we are reprogramming all donor cells. So just look at the right iPSC clone, the one that has less mutation, less deleterious mutation. And interestingly, so the very old donors actually have fewer mutation than expected. So for the second part of my talk, I want to focus on another topic. So we want to do disease modeling with iPSC. And we know that monogenic disease in general are very rare. So what we are going to face is just the genetic risk for disease. So this is just a very crowded map from GWAS catalog of MBL. These are all the single nucleotide polymorphism that have been associated with disease. So this is what we are going to face if we want to do disease modeling of common diseases. And also, we cannot study only diseases that are related to gene. Most of these polymorphisms are in non-coding region and non-in gene. So if we do a very simple diagram of human genetic variation and diseases, so we know that we have two factors. One will be the frequency in the population, and the other one is the impact on disease risk. So usually common variants are very low impact for disease risk, whereas rare variants usually have a more high impact, like monogenic disease. So it's hard to find a kind of a good balance to make a good model to study risk disease. So, and I think we found it in the 9p21.3 cardiovascular disease risk haplotype. It's a very interesting part of the genome. It has high prevalence and high impact. Is human-specific, is present in 50% of non-African population, so this is a very high prevalence. And as high impact, it can double the risk of getting cardiovascular disease in homozygous risk. So it's quite robust. So it's in chromosome 9 and is an interesting region because it's a very large 60 kb of the genome. It has no coding gene, so it's a non-coding region. It has only about 10 exons of a long non-coding RNA and some enhancers. So what we like about it is just that it's human-specific, and his correlation with cardiovascular disease is very strong. So it has been reported in many GWAS studies and many scientific reports. So it's a very well-established link. So what is... this locus is present in two different flavor, risk and non-risk. So what is different between risk and non-risk people? It's basically about 100 SNPs, so it's not only one causative single nucleotide polymorphism, it has many, and we don't know actually if one of those is causative. So how do you really study something like that? You cannot really change one base. So we thought about just knocking it out. So completely, a complete deletion of the 60 kb, and of course it's a kind of challenging, but we tried. And what we did was like recruiting, of course, the right samples. Again, we collaborate with Eric Topol and Ali Torkamani. So we took blood from donors that are homozygous risk or homozygous non-risk. Take the PBMC and reprogram in iPSC. And then with a nice collaboration with Sangamo, we tried TALEN nucleases. So with TALEN, we used two independent set of TALENs, and we were able to have wild type and knockout lines, completely knockout for the 60 kb locus. So in these, with quite a good efficiency, we got heterozygous deletion and knockout deletion in both patients with both TALEN set. So the first thing we did was, of course, looking at the iPSC. So we took our isogenic line with all our genotypes and just did pluripotency marker immunohistochemistry. And as you can see, the cells have no major defect, at least in expressing marker of pluripotency. And this is just the whole transcriptome that show you that the cells are not really clustering by the knockout of the locus, but they're just clustering by patient of origin. So we can say that the deletion of the locus is not really impacting the iPSC function, and this is good because we can go ahead and actually do more interesting thing. So, of course, using iPSC is great because we can differentiate them in any sort of tissue, any kind of cell type. And we are talking about cardiovascular disease. So we want to just study cells that are relevant for the vasculature. And we focused on vascular smooth muscle cells. We applied this protocol that makes mesodermal progenitor first and then smooth muscle cells. As you can appreciate, cells, all 4 genotypes can actually make mesodermal progenitor and also smooth muscle cell at the end. So we went ahead and did RNA-seq from these 2 steps, mesodermal progenitors and smooth muscle cells. And for the mesodermal progenitors, we did RNA-seq and do just differentially expressed genes. And the first comparison we did was like non-risk, and the isogenic knockout line. We found a very minimal difference. Only 3 genes were differentially expressed. But if you go to the risk, so risk cells versus their isogenic knockout line, so they have increased in number of genes that are differentially expressed. And I think you can appreciate from this heat map that the risk cells look very, very different than the other 3 genotypes, even the knockout of the risk. So if these are our 4 lines, basically the knockout risk look more similar to the non-risk cells. This was quite interesting, so we went ahead and looked at the vascular smooth muscle cell stage. Again, the first comparison was for the non-risk and knockout, only 87 genes. So more than 3,000 genes are differentially expressed just by knocking out the risk allele. And if you look at the principal component analysis, it's pretty clear that the risk cells are totally different than the other 3 genotypes. These knockouts look more similar to different genotype than the risk. So, and these 3,000 genes are all over the genome. We didn't find really an enrichment in one particular region. And by doing just the gene ontology analysis, we asked, what are these 3,000 genes? We found very interesting categories and relevant actually for the muscle. We found genes that are differentially expressed for muscle contraction and development and also biological adhesion. So we wanted to see if these transcriptional changes were actually functional and the smooth muscle cells were actually impacted functionally. So we had a collaboration with Dr. Adam Engler at the Sanford Consortium, and we did adhesion assay, 2 independent adhesion assay to assay smooth muscle cells. And as you can see in both of them, the risk cells are less adhesive than the other 3 genotypes. And knocking out the locus actually can rescue this defect. So we tried also contraction assay, and as you can see in this graph, the cells that are risk carrier are well less contractile. So, but these actually can be rescued if you do some pharmacology and you use drugs like bradykinin that can increase contraction. We are able to rescue the cells, meaning these cells are able to contract. So we can actually test drugs for that. So this was very exciting, I think, but the problem is we used isogenic lines that are a very clean way to study a phenotype, but just from 2 donors. So of course we want to see if our phenotype is actually consistent. So we took other 2 non-risk, and 3 risk donors. We did iPSC, smooth muscle cells from them, and then checked for some adhesion molecules, genes that we took from our RNA-seq, and indeed would confirm the same trend. And also the same trend is also true for the adhesion assay. So when you have risk cells, these are less adhesive. So to conclude this second part, I would say, so we were able to use genome editing to delete a very large portion of the genome, 60 kb in iPSC. And we used isogenic line that I think is a very nice approach. And for us, it actually was really good to discover a new function of the 9p21.3 that was never shown before. And the 9p21.3 risk affect adhesion and contraction of smooth muscle cells, so this can be relevant actually for the disease. And of course, we can use these lines for doing drug screening, and it will be nice to find compounds that can actually rescue the defect we see. And I will conclude just to tell you that our iPSC are well characterized, and you can find them at YCell. We have two collections, the elderly and the coronary artery disease, so they're available for anyone to use. And I will thank you, and thank you my lab members, and in particular people that are working on this project with me, like Pavel and Mike and Will, and of course our collaborators, Eric Topol, Ali Turkamani, Adam Engler, and Fyodor Urnov. Thank you.
What we'd like to do is take one or two pressing questions, and then for each speaker, and then at the end we hope to have 15 or 20 minutes left to have a panel where we can ask more general questions. So any pressing questions for Dr. Lasorda? Yes?
What is your speculation on the lower mutation rate in the centenarians in their IPS studies? You left that question open somehow. I promised to answer that.
Oh, yeah, sure. I mean, this is it's also already published, so maybe you find more detail in that. But it's interesting because what we found is that cells that are from old donor express less of TET genes. So basically they have a resistance to demethylation because they have less expression of enzymes that are like responsible for demethylation. And also, I think what happened is that in the old donor, we have a restricted pool of cells in the blood. There are probably hematopoietic stem cells that divide less, so they acquire less mutation, and they will be available for reprogramming. And what happened is that if you have a pool of cells that have less mutation that is available for reprogramming, you're going to end up with iPSC with less mutation. And actually, this was the case. This was our model. So in the study, we tried to find a way to prove it by doing some kind of analysis on the type of mutation we can find. But yeah, the bottom line is that they have this restricted pool of hematopoietic stem cell available.
Great, thank you. We'll follow up in the discussion. Thanks again.